Hello everyone, welcome to the channel Microbial World. In this video, we will be learning about Salmonella typhi. Salmonella belongs to the family Enterobacteriaceae. The isolates that are responsible for human gastrointestinal diseases belong to the species Salmonella enterica. More than 2,500 unique O serotypes are present. The serotypes of Salmonella enterica are Salmonella typhi murium, Salmonella paratyphi, and Salmonella enteritidis. Salmonella typhi murium, the causative agent of typhoid fever, is the most important member of this genera. Typhoid bacillus was first observed by Eberth in 1880 in the mesenteric node and spleen of fetal cases of typhoid fever, and it was later on isolated by Gafke in 1884. It came to be known as Eberth-Gafke bacillus or Eberthella typhi. In 1885, Salmon and Smith described the bacillus and redesignated it as Salmonella typhi. They are divided into two groups, enteric fever group and food poisoning group. Enteric fever group consists of typhoid and paratyphoid bacilli. They are human parasites. Food poisoning group are animal parasites, but they can also infect human beings, producing gastroenteritis, septicemia, and localized infection. Morphology. Salmonella are gram-negative rods, so they appear as pink or red under the microscope. This is due to their cell wall structure, which has a thin peptidoglycan layer and an outer membrane containing lipopolysaccharide. They measure 2 to 4 into 0.6 micromuum. They are non-acid-fast bacteria, which means they do not have the waxy lipid-rich cell wall that is a characteristic of acid-fast bacteria. They are non-capsulated, so they do not form a polysaccharide capsule around the cell wall. And they are non-sporing, so they do not produce bacterial endospores. They are motile with peritrichous flagella. Peritrichous flagella means multiple flagella will be distributed around the cell surface, and this numerous flagella will help the bacteria in its movement. Salmonella gallinarum and Salmonella pullorum are non-motile. They produce type 1 fimbriae. Fimbriae are hair-like appendages found on the surface of many gram-negative bacteria. Fimbriae enables bacteria to attach to epithelial cells, particularly in the urinary and gastrointestinal tract. Antigenic structure. Salmonella possess the following antigens: flagellar antigen, somatic antigen, and surface antigen. Flagellar antigen or H antigen. As the name says, it is a part of the bacterial flagella. It is heat labile and alcohol labile. So when we say it is heat labile, it means that it can be destroyed when exposed to high temperature, and it is also alcohol sensitive. Salmonella exhibits variations, which means it can switch between different H antigens, and it is referred to as phase 1 and phase 2 antigens. O antigen or somatic antigen. It is a heat-stable polysaccharide located on the outer membrane of the bacterial cell wall. It is a part of the lipopolysaccharide complex, and it determines the serogroup of Salmonella strain. O antigen can be extracted from the bacterial cell by treating with trichloroacetic acid. This was shown by Boivin, and therefore it is also known as Boivin antigen. VI antigen or surface antigen. It is an acidic polysaccharide capsule antigen found in some strains of Salmonella, mainly in Salmonella typhi and Salmonella paratyphi C. It enhances virulence by protecting the bacteria from host immune response. So when we say it acts as a virulent factor, it means that it inhibits phagocytosis, resists complement activation, and bacterial lysis by alternative pathway. It is heat labile but alcohol stable. Cultural characteristics. Salmonella is an aerobe and facultative anaerobe. Aerobe means they can grow in the presence of oxygen. Facultative anaerobe means they can grow in the presence and absence of oxygen. They can grow at a range of pH 6 to 8 and optimum temperature 37 degrees Celsius. Now on nutrient agar and blood agar, The colonies are large, moist, grey-white, circular disc, smooth convex surface with an entire edge. The size and degree can vary depending on the serotype. On deoxycholate citrate agar, the colonies are pale or nearly colorless, smooth, shiny and translucent. Sometimes they have a black center and sometimes they are surrounded by a zone of clear medium. On McConkey agar, the colonies are pale yellow or nearly colorless. Salmonella appears colorless because it does not ferment lactose. On Wilson-Blair bismuth sulfite agar, colonies appear green or pale brown. Large discrete colonies have a black center and clear edge. On Psilocybin deoxycholate agar, the colonies appear red with black center. This is because Salmonella produce hydrogen sulfide that react with ferric ammonium citrate to produce a black center. Pathogenesis. Salmonella is ingested through contaminated food or water. The common sources are undercooked poultry, meat or eggs. Once they are ingested, it survives the acidic environment of the stomach and passes through the stomach and reaches the small intestine. In the small intestine, it attaches to the intestinal epithelial cells. Now, how is this attachment carried out? It attached to the small intestine mucosal membrane using adhesins such as fimbriae, pili or outer membrane proteins. Once they are attached, they invade into microfold cells or M cells in Peyer's patches and also enterocytes. Now these microfold cells are specialized epithelial cells found in the lining of the intestine, mainly in Peyer's patches. And what are Peyer's patches? These are organized lymphoid nodules in the small intestine. And they also invade into enterocytes, that is columnar epithelial cells. So, it invades into the M cells in Peyer's patches and also enterocytes. Now, once they are inside the host cell, they reside in a specialized compartment known as Salmonella containing vacuole. In this vacuole, they are protected from lysosomal degradation and also they are provided with a favorable environment to replicate. So, they multiply in the endocytic vacuole and they are transported across the cytoplasm and get released into the blood. And through the blood they can disseminate to other body sites and cause systemic infection. Virulence factors. Now, in order to regulate the attachment, engulfment and replication, there are two clusters of genes on pathogenicity island 1 and 2. Now, what are these Salmonella pathogenicity islands? These are genomic regions in pathogenic bacteria that contain a cluster of genes that encode virulence factors. Salmonella pathogenicity island 1 and Salmonella pathogenicity island 2 are very important. Now, pathogenicity island 1 encodes Salmonella secreted invasion proteins and type 3 secretion system. This helps to inject proteins inside the host cell. Pathogenicity island 2 encodes a second type 3 secretion system, that is type 3 secretion system 2. This helps Salmonella to survive and replicate within the host cell vacuoles by modifying the host cell environment. An inflammatory response mediates the release of prostaglandins and triggers cyclic AMP, which causes hyperfluid secretion. Hyperfluid secretion is the excessive secretion of fluid into gastrointestinal tract that can lead to diarrhea. Clinical manifestations. Gastroenteritis. It is caused by non-typhoidal salmonella such as Salmonella typhi and Salmonella enteritidis. Gastroenteritis is the inflammation of intestinal tract. It is a common form of salmonellosis. Incubation period is 6 to 48 hours. It is acquired through the consumption of Salmonella contaminated food or water. Symptoms include nausea, vomiting, non-bloody diarrhea, headache, abdominal cramp, fever and myalgia. The symptoms can persist for 2 to 7 days. Septicemia. All species of Salmonella can cause bacteremia. Common species include Salmonella typhi, Salmonella paratyphi and Salmonella cholera suis. There are higher chances of infection in pediatric, that is young children, geriatric, old people, and immunocompromised patients, those suffering from sickle cell disease or HIV infection. Septicemia can result in septic shock and multiple organ failure. It can cause localized suppurative infections, that is, the formation of an abscess or pus. For example, endocarditis. It is the infection of inner lining of heart chamber and heart walls known as endocardium. Osteomyelitis is the infection or inflammation of bone, and arthritis is the inflammation of joints. Enteric fever. It is also known as typhoid fever. It is caused by Salmonella typhi and Salmonella paratyphi. The bacteria pass through the intestine cell lining and it is internalized by macrophages. They multiply after reaching the spleen, liver, and bone marrow. Incubation period is 1 to 2 weeks. Symptoms include slowly increasing fever, anorexia. Anorexia is an eating disorder. It is the lack or loss of appetite for food. Malaise means weakness, headache, myalgia, rose spots. Rose spots are faint maculopapular rash that is seen in some patients. Hepatosplenomegaly, which is the enlargement of liver and spleen. Bradycardia means slower heart rate. The symptoms can persist for 1 week or longer. Asymptomatic colonization. Strains of Salmonella species can remain in the patients even after recovering from enteric fever. Chronic colonization of Salmonella species can occur for more than 1 year, which means chronic carriers can harbor the bacteria in their gallbladder and shed it intermittently in their stools without showing any symptoms. Lab diagnosis. Stool culture is the most common method for diagnosing non-typhoidal Salmonella gastroenteritis. The stool samples are cultured on media such as Xylose lysine deoxycholate agar or Salmonella shigella agar to isolate Salmonella species. Blood sample is essential for diagnosing typhoid fever and Salmonella bacteremia. Blood cultures are performed to detect the presence of Salmonella in bloodstream. Urine culture may be positive in case of typhoid fever. Bone marrow culture is a highly sensitive method for detecting Salmonella typhi, particularly in patients who have received prior antibiotic treatment. Widal test is the most common method for diagnosing Salmonella infections. It measures antibodies against O and H antigens of Salmonella typhi. A significant rise in antibody titers supports the diagnosis. PCR is another highly specific and sensitive method for detecting Salmonella DNA in clinical samples such as blood, urine, or stool samples. It provides rapid results and it is useful in cases where there is low bacterial load or prior antibiotic treatment. Rapid diagnostic test. Immunochromatographic test provides quick results. It is particularly useful in resource-limited settings. Enzyme-linked immunosorbent assay ELISA detects Salmonella antigens or antibodies in blood or stool samples. Ultrasound or CT scan can be used in complicated cases of typhoid fever to detect abscesses, hepatosplenomegaly, or intestinal perforation. Treatment. For gastroenteritis, fluid and electrolyte replacement can be used. Antimicrobial treatment is also useful to prevent the systemic spread of infection. Chloramphenicol, ampicillin, trimethoprim sulfonamide, cephalosporins, ceftriaxone, and quinolones, norfloxacin and ciprofloxacin are effective. Typhoid vaccines are also available. Oral TY21A vaccine is a live attenuated vaccine that is given as a series of capsules. Injectable VI polysaccharide vaccine is an inactivated vaccine that is given as a single injection. Thank you for watching.